Hello everyone and welcome to ASAP Northwood, where we take boring scientific stuff and make it sound less boring. Probably. Maybe. I don't know. So our question for today is, what is resting membrane potential? Let's answer that, shall we? Basically, resting membrane potential is the state where a neuron cell has a charge of 0 millivolts outside of the cell membrane and a charge of negative 70 millivolts inside the cell membrane. But how is this unequal electric charge maintained? Inside of the cell membrane is a high concentration of potassium, the Ks, and a high concentration of sodium, the Nas, outside the cell membrane. Throughout the cell membrane are NAK pumps powered by ATP that pumps 3 Nas out for every 2 Ks pumped in. Throughout the neuron, with a high concentration of potassium inside the cell and a high concentration of sodium outside the cell, there are multiple leakage channels that allow large amounts of potassium to leak out, but only a small amount of sodium to leak in. An AK pump that forces 3 sodium ions out for every 2 potassium ions in, combined with the multiple potassium leakage channels leading out and a smaller number of sodium channels leaking in, leads to a higher concentration of positive ions inside the cell than out, making the inside negative compared to the outside. A nerve impulse is essentially when information is being transmitted from one neuron to another through the use of electrochemical signals. This information pathway goes through several stages. First is depolarization. Sodium ions flood into the cell membrane through the use of sodium voltage-gated channels. Then it's repolarization. This time, potassium floods out of the cell through potassium voltage-gated channels. Hyperpolarization is basically when too much potassium floods out of the cell before returning to rest, closing the voltage channels and relying on the NAK pumps and leakage channels to maintain RMP. This entire process is called propagation. Going back to the neuron, starting with resting membrane potential, sodium floods into the neuron, causing nearby sodium voltage-gated channels to open in rapid succession down the entire axon. Anyways, as information passes from neuron to neuron, the electrochemical signals reach the axon terminal, ending at a synapse. There, vesicles containing tiny neurotransmitters are released into the synapse and either propagate through another neuron or stimulate an effector. Whether it's a muscle that contracts or a gland that releases hormones. So in conclusion, a nerve impulse goes through several stages in its pretty short lifespan, starting at negative 70 millivolts at rest, gaining sodium ions to reach 30 millivolts in depolarization, losing potassium ions to go back to negative 70 millivolts in repolarization, losing some more potassium in hyperpolarization, before finally going back to negative 70 millivolts when returning to rest. Thanks for watching, everyone. Aren't those guys awesome?